Hello viewers, today we are going to talk about the plant hormone gibberellin, which is the main regulator of elongation of the stem. Now, gibberellins are a family of terpenoid compounds made of isoprene units. They have a basic ring structure which is called end corine, and all the members of the family are structurally related. Now, there are several gibberellins isolated, more than 100 from different plant species as well as bacteria. All these compounds are not bioactive. Only few are active within plant. For example, GA1 and GA4. These other forms are either metabolic intermediates or precursors or inactive degraded forms. So from now onwards, if we mention gibberellin, we will be meaning GA1. Within plant, gibberellins may also be glycosylated to give either an inactivated form or a storage form. If we exogenously add gibberellin to a dwarf plant, then it shows dramatic elongation of the stem. It becomes as long as the wild type. But interestingly, if we add gibberellin to a wild type plant and expect that it will grow very tall, that doesn't happen. This is because endogenous gibberellin present within the wild type plant has a feedback effect to check that the plant doesn't become over long, because that is not very healthy for the plant. There are plant forms called rosette forms. For example, cabbage in short days take up that rosette form. If we apply gibberellin to rosette plants, then it also shows dramatic elongation. These two facts indicate that exogenous gibberellin can stimulate stem elongation in dwarf as well as rosette plants. Now, this picture shows two corn ears, both coming from dwarf mutants. Now, in the lower picture, we see conspicuous anthers have developed. This is because this lacks gibberellin. But the upper corn ear doesn't show this, and it shows suppression of stamen development because gibberellin has been applied to this. Thus, gibberellin can also regulate sex determination. Now, this picture shows two different types of grapes. These are Thompson's seedless grapes. The left one without gibberellin treatment and the right one with gibberellin treatment. And the gibberellin treated grapes shows much higher increase in number as well as size of the fruit. The gibberellin can also regulate fruit production. Now, this is a metabolic pathway of gibberellin biosynthesis as well as degradation. If we look at this, we can identify three critically important enzymes. Two of them are involved in gibberellin biosynthesis, the active form, that is GA1, and another one is involved in inactivation of the active form, that is its degradation. These are GA20 oxidase and GA3 oxidase that promote formation of GA1, and another is GA2 oxidase that promotes its degradation. Now, to locate the sites of gibberellin synthesis in plant, a reporter gene based assay system was developed where the luciferase reporter gene is linked with GA20 oxidase promoter. Then this was sprayed with luciferin, and the color that is developed was taken in a picture, and the color intensities were analyzed, and then it was modified with false color so that we can identify which part expresses the gibberellin most. This shows that the maximum level of gibberellin biosynthesis happens in the young developing leaves and the shoot apex. Consequently, gibberellins are mostly biosynthesized in the apical tissues of the plant. Now, this is a northern blot experiment to check the levels of the mRNAs for the enzymes of gibberellin biosynthesis as well as degradation. Two kinds of plants are taken. One is a dwarf variety which doesn't have gibberellin, that is gibberellin deficient, and another is a wild type that has normal gibberellin level. Now, the northern blot image indicates that the mRNAs for gibberellin biosynthesis is highly expressed in the gibberellin deficient dwarf plant, whereas it is much less expressed in the wild type plant having gibberellin. On the other hand, GA2 oxidase, that is the degradative enzyme, is much less expressed in the dwarf plant and much highly expressed in the wild type plant. This indicates that gibberellin can regulate its own biosynthesis by a feedback mechanism. Light, the most important environmental factor for plant, can influence gibberellin biosynthesis as well as sensitivity. The left graph with pea seedlings shows that in dark there is moderate level of gibberellin synthesis. When this is exposed to light, there is an immediate and rapid decline in the gibberellin level. This low level continues for about 1 day, and then onwards there is a dramatic increase in the gibberellin level. The right panel shows the effect of exogenous gibberellin on dwarf gibberellin deficient plants either in dark or in light. Here we see that the rate of elongation in dark is quite high in presence of gibberellin. When this is shifted to light, then there is a significant decline in the rate of elongation. This indicates that though light promotes gibberellin synthesis, but it decreases the sensitivity of plant towards gibberellin. This is also related to checking overgrowth of plants. The mechanism is mediated by light-induced phytochrome mediated signaling pathways. Photoperiod is another environmental factor important in this context. Photoperiod means either short days or long days. Now in short days, the gibberellin level is low, and in long days, the gibberellin level is high. Now spinach in short days take a rosette form. In long days, it takes the normal long form. Now if we apply a gibberellin biosynthesis inhibitor, then even in long day we can see that the plant has adopted the rosette form. But now to this, if we add exogenous gibberellin, then again it takes the normal long form. So this data shows the increase in the level of gibberellin with increase in the number of long days exposed. From this we can see that there is a huge increase in the gibberellin molecules that are metabolic intermediates or precursors or inactive or degraded forms. The increase of the bioactive GA1 is moderate, but even this moderate increase is sufficient to exert the stem elongation effect that we have observed. The photoperiod can also regulate formation of tuber in potatoes via change in the level of gibberellin. Short days decrease the level of gibberellin, induces tuber formation in potatoes. In long days, when the gibberellin level is high, there is no potato tuber formation. Another plant hormone, that is auxin, plays a very important role to regulate the level of bioactive gibberellin in plants. In the left panel, we can see that the gibberellin level rapidly declines after the plant is decapitated. That means the source of auxin is removed. But when we exogenously apply auxin to this, then the gibberellin level goes up very high. This northern blot experiments shows the level of mRNA expression upon auxin induction. So we compare the mRNA levels of GA3 oxidase that promotes gibberellin biosynthesis versus GA2 oxidase that promotes degradation of gibberellin level. Here we can see that when there is auxin, then the biosynthetic genes are highly expressed. When there is no auxin, then the expression level goes down. On the other hand, in presence of auxin, the expression of the degradative enzyme is very, very low, whereas in its absence, it is very high. And the change in the level of the biosynthetic genes is proportional to the time of exposure to auxin. Thus, auxin activates transcription of the biosynthetic genes and suppresses the transcription of the degradative genes. So, auxin after synthesis in the apex is transported basipetally to the internode, where it promotes the biosynthesis of gibberellin and inhibits the degradation of gibberellin. Thereby, there is an increase in the gibberellin level in the internode, resulting in the growth of that region. Now, this figure summarizes all the different internal and the environmental factors that regulate the level of gibberellin in plant. For example, photoperiod and red light, they promote gibberellin biosynthesis. On the other hand, the internal factor auxin also promotes gibberellin biosynthesis. These two factors inhibit gibberellin degradation. There is products of the gibberellin signaling pathway or gibberellin response pathway that have a negative feedback effect on gibberellin biosynthesis and that positively regulates gibberellin degradation. The major mechanism of gibberellin induced stem cell elongation is increasing their cell division. In this image, we can see that in the longitudinal cross sections, after application of gibberellin, there is huge increase in the number of dividing cells. Thus, gibberellin promotes cell division and thereby growth. At the level of cell cycle, gibberellin promotes transition from G1 to S. More cells enter S phase, thereby more cells will enter the G2 phase, and after that more cells will enter the M or mitotic phase. Consequently, number of dividing cells increases. Gibberellin does this by upregulating the cyclin-dependent kinases that promote the transition from G1 to S phase. Now, several negative regulators have been identified that downregulates the transcription of the gibberellin-induced genes. These negative regulators are SPY, RGA and GAI. All are proteins. Here, RGA and GAI, they are transcriptional repressors of the gibberellin-induced genes. SPY, on the other hand, is a N-acetyl glucosamine transferase that can transfer the N-acetyl glucosamine moiety to an important serine residues present on these transcriptional repressors and thereby activate them. Now, in absence of gibberellin, SPY is active. It then activates RGA and GAI, which then transcriptionally repress the gibberellin-induced genes and growth of the plant is suppressed. But in presence of gibberellin, activates the receptor and then the activated signaling pathway can directly inactivate SPY, GAI and RGA. Consequently, the transcriptional repression of the GA-induced genes are lifted, these genes are then expressed and we see the result, that is elongation of the stem. Another very important physiological effect of gibberellin is promoting germination in seed. Here we see a cross-sectional view of a barley grain where gibberellins are produced in the embryo. From the embryo, it diffuses into the endosperm via the scutellum, and from the endosperm, it goes into the aleuron cells and enters the cells. There, gibberellin induces the expression of several hydrolytic enzymes such as alpha amylase, protease, DNase, RNase, etc. These enzymes are then secreted into the endosperm where they hydrolyze the macromolecules and produce soluble nutrients. These soluble nutrients can then re-enter the embryo and thereby promote the growth of the embryo. This image of the aleuron cells, in panel A, we can see the cells, the nucleus and the protein storage vesicles. Panel B and C shows the aleuron cell protoplasts. Panel B is the early stage of production of the hydrolytic enzymes where the protein storage vesicles are quite small. Panel C shows the late stage where the protein storage vesicles have been enormously enlarged because now they are holding all the overexpressed hydrolyzing enzymes which will be secreted. Upon gibberellin application, there is a dramatic increase in the enzyme level, particularly alpha amylase, RNase, DNase, protease, etc. in the aleuron cells. This is preceded by a parallel increase of mRNA for these enzymes. Thus, gibberellin transcriptionally activates the genes for these hydrolyzing enzymes. Gibberellin causes increase in the expression of transcriptional activator GAMIB. This GAMIB activates the hydrolyzing enzyme genes. So, the expression of GAMIB precedes the expression of the genes of the hydrolyzing enzymes. Gibberellin can activate multiple signal transduction pathway in the aleuron cells. Some of them are calcium independent, some of them are calcium dependent. The calcium independent pathway, for example, involves cyclic GMP, GAMIB, and the result is overexpression of the hydrolyzing enzymes. The calcium dependent pathway involves calmodulin and other members. These are involved in secretion of the enzymes. Several other regulatory factors have been identified, and combining them, a signal transduction pathway of gibberellin-induced germination has been proposed. This indicates that when gibberellin binds to its cell surface receptors, it triggers calcium-dependent and independent cascades. In the calcium-independent cascades, G proteins are activated with the resulting production of cyclic GMP that then activates some downstream regulatory molecules which can enter the nucleus, and it can bind to a transcriptional repressor called DELA. This DELA repressor represses the transcription of GAMIB. When the signaling molecule binds to DELA, it is inactivated and dissociates from the gene. This allows the expression of the GAMIB gene. The mRNA that is transcribed goes to the cytoplasm where it is translated, and the GAMIB transcription factor then re-enters the nucleus where it binds and activates the hydrolyzing enzyme genes such as alpha amylase, etc. The resulting mRNA is translated in the rough endoplasmic reticulum, and the enzymes are produced in secretory vesicles. Now, the parallelly activated calcium dependent pathway involving calmodulin is involved in the secretion of these enzymes via the secretory vesicles. Now, depending on the various physiological effects of gibberellin, several bioassay techniques have been developed. For example, measurement of the elongation of gibberellin stem induced by gibberellin. We can also use the gibberellin-induced seed germination. Actually, this is more accurate method for gibberellin bioassay. For example, we can take seeds from dwarf pea plants and treat it with our test sample that may contain gibberellin. We also put it in water as a negative control. Now, if gibberellin is present, then in about 5 days there will be germination and development of the coleoptile, and we can measure the length of the epicotyle. The length of the epicotyle is proportional to the amount of gibberellin present in the test sample. Also, gibberellin induces hydrolysis of starch resulting in production of reducing sugars. So, measurement of reducing sugars in the endosperm is also a good bioassay method. So, the test sample, if contains gibberellin, it will increase the level of reducing sugars in the endosperm, and we can measure it, and the quantity of reducing sugars present is proportional to the amount of gibberellin present in the test sample. Now, gibberellin has innumerable applications. For example, it is used to induce the increase in number of fruit as well as the size of the fruit. For example, in grape, in tomato, as well as in apple. If applied to the unpollinated flowers, then gibberellin can also induce formation of seedless fruit, that is parthenocarpic fruits. Gibberellin increases the yield of malt in barley grains. It is also used to break dormancy of seed and bud in tobacco and lettuce. Gibberellin also delays senescence and ripening of fruits, particularly citrus fruits. So, it is used to increase the marketing period as well as the storage period of these fruits. Off-season flowering can also be induced by gibberellin, particularly for long day plants. Application of gibberellin to sugar canes increases their length as well as their yield to a very high level. And finally, gibberellin is used to induce maturity in juvenile conifers. This results in early production of seeds. So viewers, today we have discussed the different aspects of the plant hormone gibberellin. For example, the different types, the structures, the bioactive forms, the inactive forms, the biosynthetic and degradation metabolic pathway, the physiological effects, the different bioassay techniques, the different applications of gibberellin, as well as the molecular mechanism. Thank you.